Today we'll be discussing the properties of the Watson and Crick DNA model. DNA is a double helix made up of stacked paired nucleotides. Each individual nucleotide is composed of a ribose sugar, a nitrogenous base, and a phosphate. First, we will discuss the properties of the ribose sugar attached to each nucleotide. Each carbon on the sugar is designated with a number 1 through 5 prime. The 1' carbon is where the base attaches. The 2' carbon distinguishes whether it's RNA or DNA nucleotide. For example, if there's an OH attached to the 2', it's a ribose, or RNA. If there's only an H attached, it's deoxyribose, therefore DNA. The 3' end joins the 5' end of the incoming nucleotide, therefore bonding to the next 5' phosphate. The 5' carbon is where the phosphate group attaches to. Next, we will discuss nitrogenous bases. There are 4 bases in DNA: adenine, thymine, cytosine, and guanine. Each is abbreviated A, T, C, and G respectively. Each base has a unique pair. A's paired with T's, C's paired with G's. In addition, A's and G's are considered purines as they have a double ring structure, and C's and T's are considered pyrimidines as they have a single ring structure. These base pairs are held together with hydrogen bonds, and although hydrogen bonds keep the bases paired, it's not the main force joining the 2 strands of DNA together, and we will discuss the main force later in the video. We will discuss the phosphate backbone. It is connected by phosphodiester bonds and has a negative charge and is on the outside of the DNA while the bases are on the inside. As the phosphates have a negative charge, they like water and are considered hydrophilic. However, the bases are hydrophobic and hate water. The bases stack or huddle together in order to keep the water out. These hydrophobic interactions are the main force stabilizing the DNA, or double helix. And you can think of the bases as a group of people who hate the cold and huddle together to keep the cold out. So the closer they are together, the more the cold is kept out. Looking at the big picture, the Watson and Crick model is the B form of DNA where there are 10 bases per turn and it's right-handed. You can tell the DNA is right-handed as the spiral goes up and to the right. The strands are also anti-parallel to one another, and this means if one strand goes in the 5' to 3' direction, the other will go in the 3' to 5' direction. If you know the base sequence of one strand, you also know the complementary sequence of the other. DNA also has major and minor grooves. The large gap is a major groove and the small one's a minor groove. The grooves are important in order to allow proteins to interact with the bases. Looking at an example problem, we will discuss how the midpoint of thermal denaturization, or TM, changes in the DNA. In order to separate the antiparallel strands, DNA must be denatured where hydrogen bonds and hydrophobic interactions are broken down with heat. However, the closer the DNA strands are together, the more stable it is, and the more heat is required to separate them. Therefore, when the negative charges of the 2 phosphate backbones are partially neutralized and not repelling each other as much, the bases can huddle even closer together and further stabilize the DNA strands. That means that the TM of the DNA would increase as more heat would be required to denature the further stabilized DNA. Here are my references. Thank you.